The problem is those initial DNA methylation clocks, they're great at telling you about your chronological age, predicting what your age is, but they're kind of lousy at telling you about your future risk or telling you about your risk for morbidity, clinical information, or mortality.
Can you actually, before we go further, why is there a good reason that DNA methylation tracks with age or predicts age? Does our DNA tend to become hypermethylated over time or hypomethylated over time?
There are different theories. Someone who's written a lot about this in a very intriguing way is David Sinclair at Harvard. He has his entire information theory of aging, he calls it, which is largely boils down to the fact that aging entails basically a loss of epigenetic information over time. So DNA methylation, which are these chemical marks on top of DNA, aren't static across life, and they're certainly not static or consistent across cells in your body. And so what they are is very dynamic, and he has some compelling research showing that, at least in some of the studies that he's worked on, some of these epigenetic regulatory mechanisms sort of will have dual or shared roles where they might be involved both in epigenetic regulation, but then also DNA damage repair and somatic maintenance and things like that. And as time, when they have to move back and forth between these roles, they get lost and you tend to lose information. So the theory is that epigenetic regulators over time will change in some kind of stable way in terms of thinking about them through time, and that tracks really well with age. So, in some cases, for methylation, the analogy of a clock is apt because it just seems to kind of tick along with time. And so that's why it's so good at capturing chronological age information. The problem is that there are so many steps that have to go from DNA methylation to the end result, which would actually influence a phenotype we care about, which is basically changing protein levels, that those methylation marks don't always correlate strongly with ultimately the protein, either for the gene that methylation mark is in or something more downstream or distal, that there tends to be this disconnect